All right, we've looked at mitosis. Now let's take a look at meiosis. In meiosis, remember the goal is to make haploid cells that are, at least in animals, going to become the gametes. And meiosis is a bit more complicated than mitosis because it involves two divisions. The first division is called meiosis 1, and you get two daughter cells from that. And then each of those two daughter cells will undergo a second division, which is called meiosis 2. And each of those divisions has its own prophase, metaphase, anaphase, and telophase. And we're going to kind of simplify the situation a little bit, and again, not get caught up on everything that goes on, but we're going to worry about the behavior of chromosomes, because the behavior of chromosomes during meiosis goes a long way to explain the patterns that we see when we look at inheritance. Remember, and that comes about because, after all, the genes are located along the chromosomes. So let's start right in. Now, just like in mitosis, we start out with a cell that has unduplicated chromosomes, and I start out with the same 4 chromosomes that I started out when I looked at mitosis. And this is a diploid cell because it has pairs of homologous chromosomes. Here I have 2 big chromosomes. Those are the same size and shape. They are homologous. I have a smaller pair. They are the same size and shape. So I have a diploid cell. All right, now before we can begin meiosis, just like we saw in mitosis, you have to duplicate the DNA. So just like I did before, I've got my new DNA kind of hidden under the current chromosome. So I'm going to pull up the DNA a little bit here, like that, and like that. And again, I'm keeping the DNAs together because just like before we begin mitosis, the DNA molecules are attached to each other at this point at the centromere. So I end up in this situation before I even start meiosis with a cell that is diploid. It's gone through the synthesis of DNA, so each chromosome now has 2 molecules of DNA. All right, so what happens? Well, in meiosis 1, what happens first of all is that homologous chromosomes pair up. And I think I'll try to show that a little bit differently than we do here. But this one's going to pair up with this one, so they're side by side. This one's going to pair up with this one, so they're side by side. So let's see if I can get that to work here. All right. Okay, let's bring them a little bit closer because in reality they're right up against each other, kind of like that. I'm not doing a very good job here. Let's hope I can do a better job with these guys. Whoops. Here we go. Okay, so now I have 2 pairs of chromosomes, and they're physically side by side. And the reason that happens is because the DNA along the chromosomes is going to exchange with the DNA from its mate. So you're going to have new combinations of genetic material happening. And I'm not going to bother to try to show that with these chromosomes. But that's what's referred to as crossing over. You get new combinations of genes along the chromosome that did not occur in the parent cell. And we'll probably talk about that at another point in lab. Okay, so what's going to happen next is that at the end of meiosis 1, each chromosome in the pair is going to separate from its mate. So this one is going to end up separating from this one. This chromosome is going to end up separating from this one. So I'm going to go right away and show those results. Now it's not going to matter which of these 2 cells ends up with which chromosome as long as I have one big one and one small one in each cell. So let me see if I can get that to work. I'm going to have to drag these individually. So I'm going to randomly put this one down here. And I'm going to put this one over here. Notice what's happening is that each cell is ending up with 1 chromosome from each of the original pairs, like that. Okay. So notice when I'm all done, and we're ignoring the details of prophase 1, metaphase 1, anaphase 1, and telophase 1, when I'm all done, I have two daughter nuclei. First of all, since I have one of each kind of chromosome, the cells are haploid. Are the cells genetically identical? No, they're not. So, for example, the blue material here might represent genetic information that comes from your mother. The red material here and here might represent genetic information coming from your father. And notice that these two cells are not only haploid, but they are no longer genetically identical. Okay, so that's what we end up with at the end of meiosis 1. Now, each chromosome still has two molecules of DNA. So we go through meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis. And you end up with 4 cells, 2 from here, and then 2 from here. And let's see what happens. Each cell is going to get one of each kind of DNA based on size and shape. So this what will now become a chromosome ends up over here, right? The other one is going to end up in this cell. Ooh, whoops, I gotta rearrange things a little bit here, send that to the back. There we go. And that one's going to end up there. Oh, and where's this one going to go? Well, it's going to go over here, all right? Okay. Oops. Bring that to the front. There we are. So notice that now I've got two haploid cells. It may look like they're genetically identical, but in reality, because of that crossing over process, they may not be. Okay, and in fact probably aren't, because there'll be new combinations of genes along here that did not occur back here. And so what this should be represented physically is you'd have bits of blue and red interspersed, mixed up together in these chromosomes because of crossing over. So we're not showing that part of it. Now what's going to happen over here? Well, the same sort of thing. Each one of these DNA molecules is going to end up in its own cell. All right. Oops. Where's that going to go? That's going to go here. Uh-oh. I've got to bring that forward so you can see it. There we go. And this one's going to end up over here. All right. Now I've got to do the same thing. All right, there we go. So what do I have? I have 4 cells, 2 from this cell and 2 from this cell. These 4 cells are haploid. Each one has the haploid number of chromosomes, and each chromosome is an unduplicated chromosome. That is, each chromosome has just one molecule of DNA. Okay? These cells are not genetically identical to each other, and even within looking at these guys, if we were to show what happens with crossing over, these would not be genetically identical to each other either. But each cell has one of the original chromosomes that were back here, but not both from each kind of chromosome. So these cells are haploid. Now this is important in terms of genetics, because what it also implies is that for each pair of genes, remember genes in diploid organisms come in pairs because the chromosomes come in pairs. Each of these cells has one gene from each original gene pair because it has one chromosome from each of the original chromosome pairs. Okay? The other thing to remember about these cells is that in animals and in some other organisms, these are going to become the gametes, the egg and the sperm. Okay? And then in the rest of sexual reproduction, the egg and the sperm get together to give a new diploid cell, which is going to be called the zygote. All right, so remember that. All right, so this is just a brief look at meiosis, cutting away all the details of the 2 cell divisions, concentrating on what happens to the chromosomes. Remember, we start out with a diploid cell, duplicate the DNA, then we go through meiosis 1 and meiosis 2. The products are 4 haploid cells, which are genetically different from each other, and those haploid cells, at least in animals and a few other organisms, will become the eggs and the sperm. All right, so this is a quick look at meiosis. I hope you found this useful.